Dear students, today I will teach you about chromosomal alterations. In this, I will cover deletion or deficiency and duplication. First of all, what is chromosomal alteration? As you know, in a chromosome, first of all, I will tell you the structure of a chromosome. This is centromere, chromosome arm, and in this there is specific sequence of genes A, B, C, D, E, like this. And these genes are arranged in a specific sequence. And in chromosomal alteration, there is structural change of chromosomes. Either there is change in sequence of genes, or there is addition of genes, or deletion of genes. First of all, I will tell you what is deletion. What is deletion or deficiency? As you know, deletion is the loss of chromosomal segment. Loss of chromosomal segment. Deletion is of further different types. That is, in this we will study. It is of two types: terminal deletion or intercalary. Terminal deletion or intercalary deletion. In the terminal deletion, in this case, that is in the chromosome, either there is B, C, D, E, F. There is this is a pair of homologous chromosomes. And in terminal deletion, that is terminal segment is lost. That is, terminal genes are lost. AB genes, these are lost. And in this case, that is, the sequence will be, after deletion, it will be C, D, E, F. Terminal segment is lost, and the lost segment is that is AB segment is lost, and this AB segment is also acentric. If centromere is not there, no centromere. And in terminal deletion, there is only one break. There is only one break, and in case of intercalary deletion, in intercalary deletion, there is a break there is a break in the chromosome in any part. These are homologous chromosomes and genes are A, B, C, D, E, F. And in this case, suppose CD. Genes CD are removed. This portion of the chromosome is lost. If this portion of the chromosome is lost, then after the deletion, after intercalary deletion, it will be A, B, E and F. And this lost segment is CD, and in this case two breaks are there. One break is this and other break is this. When the segment is broken, this segment is broken, then again there is reunion of the broken segment. In deletion, first of all, as we are studying that we studied terminal deletion and intercalary deletion, then is what happens, what is deficiency? Deficiency is also when the loss of terminal segment is there. Due to the loss of terminal segment, the chromosomes, that is these genes, the genes are lost. That's why in the genotype there is deficiency of genes. Then is other terminology is what is deletion homozygote and other is heterozygote. When deletion occurs in the both the homologous chromosomes of a pair, then deletion is homozygous. That is, suppose this segment is lost here, and in the homologous chromosome this segment is also lost. And in deletion heterozygote, when only one of the pair of the homologous chromosome shows deletion. In this case, in this segment, that is, this is deletion heterozygote. Then as what is the cytological effect at the Peckytin stage of meiosis, at the time of meiosis, at Peckytin stage, in case of deletion, as a terminal deletion, in case of terminal deletion, that is, one arm of the chromosome is long, one pair is long and one short. Because this portion is lost. And in terminal, in intercalary deletion, chromosome, suppose CD is lost. So at the time of acquaintance phase, a loop is formed. A loop is formed. This is also called as loop or buckling effect. Now, what is the significance of deletion? What is significance of deletion? Deletion is, that is, when a gamete is formed, due to deletion, when gamete is formed, the gametes which are formed are sterile, as there is loss of number of genes. And second is, the deletion is in case of homozygote. Deletion is lethal because vital genes may be lost. In heterozygote, at the time of Pachytian stage, buccal or loop is formed, and when buccal is formed, suppose like this way, A, B, D, E, and this is C, which is lost. Here C is lost and here C is present. So at the time of pairing, the buckle is used, buckling effect is used, and this will lead to the phenomenon of pseudo dominance. Pseudo dominance means, in this case, if suppose dominant genes are removed and the recessive gene will express itself. What is pseudo dominance? In pseudo dominance, if there is loss of dominant genes due to deletion, then expression of recessive gene takes place, and which is called as pseudo dominance. So phenotypically, what are the changes occurring? First of all, in human beings, in chromosome number 5, there is a loss of 5p, this gene, and due to this, cry do catch disease is there, and in this case the child cries like a cat. In Drosophila, note winged margin is there in Drosophila. What are the effects? That is, first of all, in human chromosomes, chromosome 5, there is a deletion and the child cries like a cat, and it causes cry to cat. In Drosophila, note winged margins are there. These are also due to deletion. In human beings, one more, that is loss of third chromosome, that is genes in third chromosome. Ears are enlarged and fingers are also enlarged. In the third chromosome, enlarged ears and fingers are there. Due to chromosomal deletion in heterozygote condition, as buckling effect is there and it helps to form chromosomal map, because as we see, that is at the time of synapses, that is suppose this is A, B, C, D and E. A, B, D and E. So C is lost. So by at the time of synapses, this will locate the position of gene C. This is heterozygote, which can help in chromosomal mapping. So in deletion we have studied terminal deletion, intercalary, what is pseudodominance, what are homozygote in deletion, and what are the heterozygote. Now we will study what is duplication. In duplication, there is addition of genes. In duplication, there is addition of genes, and genes are added either to the same chromosome of homologous pair or to the other. So there are different types. In duplication, there is addition of genes. First of all, where addition takes place, first of all, either when the loss of genes, there is first of all deletion of genes, and then the deleted genes will get A, B, C, D, E, F. Suppose this segment is lost, and this will get added to the other segment, that is either at the same position or in the different position. Duplication is of different type. One is tandem duplication. Other is reverse tandem duplication. In tandem duplication, suppose the genes are A, B, C, D, C, D. That is, CD is repeating itself. And in reverse tandem, that is, the genes are repeating itself but in a reverse order. So this is called as reverse tandem. This duplication is called as reverse tandem duplication, and this is the tandem duplication. And what are the effects in duplication? Cytologically, duplication occurs either due to unequal crossing over or due to addition of genes. In unequal crossing over, that is, suppose in this arm the genes are A, B, C. A, B, C. When the break occurs like this way, here A, B and B, C, and the synapses take place between A, C, and here is A, B and again B, C. So the sequence of genes is repeated. Then what are the effects of duplication? Effect of duplications are seen in Drosophila, bar I. In Drosophila is due to repetition of chromosome 16A. And in also Drosophila, chromosome 4, there is I-less Drosophila. Then what are the effects of duplications? Duplications are, as compared to deletion, less deleterious, less harmful, and these are less harmful as compared to deletion. These duplications are also more frequent as compared to deletion. Then deletion also increases genetic redundancy. Genetic redundancy means, suppose there is gene 1, this is a repetition of gene DBC, this DB, and the effect of this gene is doubled. So this will get protected from the harmful effect of its allelic form. And genes in this case, duplications are, that is, they also help in evolution. Duplications are also help in position effect, that is locating the position of genes. because at the time of Peckitan stage, here also loop is formed. So in this lecture, in duplication, I told you about what is duplication, what are its types, that is tandem duplication, reverse tandem duplication, what are the causes of duplication, and what are the cytological effects. I also told of what are the phenotypic or genotypic changes due to duplication. In Drosophila bar I is also very important. So chromosomal alterations cover both the topic deletion and